Welcome, everybody. Thank you so much for joining us. We are doing a fireside chat today with Dr. Gregory Stein. He is the co-founder of Curtana Pharmaceuticals and became the company's chief executive officer back in 2013. During his professional career of more than 30 years, he has been a clinician and a life science executive with experience in the areas of diagnostics, medical devices, pharmaceuticals, and biotechnology. So, Greg, thank you so much for joining us today and just sharing with us a little bit about what Curtana Pharmaceuticals is and how it came to be, the work that you guys are doing right now.
Sure, thanks. Really nice to be here today. I look forward to talking with you.
So why don't we start with what Curtana Pharmaceuticals is? Why did you start the company? Why go in this direction?
So it's an interesting it's a story in terms of how it all came about, because it was total serendipity, but it was something that I couldn't pass up once I learned about it. And it all started when I met Dr. Santosh Kesari, who at the time was head of neuro-oncology at UCSD. And at the time, I had another company that was focused on something completely different than brain cancer, but I had come across some literature that suggested what we were doing might work in brain cancer. So I went up to the university to talk to Dr. Kesari about that, sort of the science around that. And in the course of the conversation, he said, "Hey, I've been working on something for the last 10 years. It started when I was at Dana-Farber with Keith Ligand and Chuck Stiles. And now I've been working on it with some colleagues here at UCSD for the last several years." And he said, "I think I have something that could really change how brain cancer is treated, but I don't really know what to do next. I don't understand that sort of the business. How do we take it from the lab to, actual drug development?" And he just wanted some feedback on this idea and some, suggestions on what to do. So I looked at it and I really liked the underlying value proposition. Now we can get into that a little later, but it really struck me how elegant it was, what they were proposing to do.
Okay.
So that was number one. And I had recently lost a dear friend to GBM. Even though I'm a physician, I'm an ER physician by training, saw many, GBM patients in the ER, either initial diagnosis or with complications. It was the first time I'd really been close to somebody and watched them go through the whole clinical course of it, and lost them to the disease. So that was really sort of fresh in my mind at the time.
So I'm curious because that kind of led into the question I was going to ask next. So as you guys were talking about this and giving him some guidance on kind of how to take this to the next step, the fact that you had a friend, the fact that you've seen this from the sidelines in your professional career, but then you had somebody close to you that you knew personally that was touched by this, how did that change the course of what you decided to do? How did that impact you wanting to really step into this with Santosh?
First and foremost, the science needed to be really compelling. But having that personal experience with it made it that much more urgent, and really, because there's lots of opportunities out there. But when I saw this and I liked the science and had just lost a friend a few months before to it, really knew how devastating the disease was. I knew how devastating it was before, but really felt it and experienced it firsthand. I don't know. It just kind of all came together. It just seemed like the right thing to do. But first and foremost, the science has to be good, because we need that compelling sense of urgency to drive us. But at the end of the day, that can't be the only thing. The science still has to be really solid, and that's what. So the combination of the two really made it a great opportunity.
And that seems to be a barrier when it comes to tackling brain cancer and finding new treatments, is that even if there is methodology that is available and scientists and doctors and researchers have an idea, the science can be very complicated and very nuanced when it comes to finding a way to
Mhm.
treat brain cancer better than we're currently treating it.
Mhm.
And so the fact that you're mentioning that the science has to be solid, because it's a huge barrier. And there are a lot of places that are like, we're not touching this because it's so complicated and it's too hard to find solid science. So tell us a little bit. You guys formed Curtana Pharmaceuticals, and the purpose of that was because what he brought to you was a potential treatment with research he had done with CT-179. So can you tell us a little bit about what that is? How does that work? What drew your attention to that? What makes it solid science?
So it's actually not too complicated to understand. So um, there's a protein called olig-2, and olig-2 is involved in normal brain development. So it's very active embryologically. So when you're still, in your mother's womb and in the first few years of life, it tells the brain to grow and to go into different cell lines and for the brain to mature. But as the brain becomes mature, it loses expression of olig-2 because it stops growing. But what happens is these gliomas, GBM being the primary one, hijacks that growth driving effect,
Okay.
and that's what turns it back on. And
Mm.
so what's interesting is that olig-2 is not in the vast majority of cells in your brain, except for some non-dividing neural stem cells. We all have stem cells in every tissue, and there's brain stem cells. But those stem cells um, transform into cancer stem cells, and they turn up back on the expression of olig-2 and some other transcription factors, and that's what just drives the growth of the tumor.
Wow.
What's interesting from a drugging perspective is because olig-2 now is just in the tumor,
Hmm.
it's not in the vast majority of cells in the brain. It's not in the bone marrow. It's not in the gut. And if we could develop a drug, this is what got me excited initially, was if we could develop a drug that was highly selective for olig-2,
Hmm.
got across the blood-brain barrier, we could hit the tumor really hard, but not kill normal brain cells and not have the normal side effects that you see with most cancer drugs that kill all rapidly dividing cells,
Right.
not just the ones in the cancer. So
Okay.
that was the real promise of this, was if we could develop a highly selective olig-2 targeting drug, we could
Right.
hit the cancer and not damage the normal brain cells and not hit the gut and whatnot. And so that's what got me excited. And he had not yet developed CT-179. He had sort of a prototype drug
Okay.
that had some decent activity, got into the brain. We took that molecule and did a whole bunch of chemistry around it. And out of that came CT-179. So yeah.
So how do you get it across the blood-brain barrier? How does CT-179, how is it introduced into the body that it can potentially get across the blood-brain barrier to get to that tumor?
Mhm. So molecules cross into the brain all the time,
Hmm.
but the brain's very selective.
Hmm.
And if you know the right properties of the molecule, you can get something into the brain.
Hmm.
Um, the problem is most drug developers who are developing for lung cancer, breast cancer, whatnot, they don't make those modifications because they don't need to get the drug into the brain.
Right.
And but it's doable. And so I hired somebody to be our VP of medicinal chemistry who had spent decades doing just that. He had worked on seizure drugs and other drugs that acted in the brain, right? 'Cause
Okay.
we give all kinds of drugs that get into the brain. It's
Yeah.
just most cancer drugs don't get into the brain. And so we just made some tweaks to it, added a methyl group here, or he did some chemistry
Yep.
blend and gave it, and we did close to 600 variations around this molecule.
Oh,
So
wow.
and it's just a iterative process. I mean, there's no magic to it. have some basic understanding of how things work. But then you just make all these different molecules and you test them in cells and see which ones get in and which ones don't. And so out of that came CT-179, which has really good drug-like properties. So it's orally bioavailable, so you can take it as a tablet.
Oh,
It stays in the body a long time, so we only have to give it once a day.
okay.
Importantly, it readily crosses the blood-brain barrier. That was really, absolutely critical, right? Because
Right.
if it doesn't get into the brain, which most cancer drugs don't,
Right.
then you're going to lose before you even get started. So that was requisite, and it does that. It gets into the brain at very high concentrations.
Okay.
So it sort of checked all the boxes from a pharmacology perspective.
Okay. So, this is fascinating. I'm curious from your experience and your knowledge base when it comes to glioblastoma and other gliomas, other cancerous gliomas in the brain, is Olig-2 something that's representative in the majority of those tumors where it's a big part of the makeup of those tumors? Or is it something that, as we're talking about clinical trials, most people understand that there are qualifiers that are needed. So you need to meet certain criteria in order to get accepted into a trial. There's certain things that can negate you from a trial. For some, it's the mutation of what the makeup of the tumor is. So is Olig-2 something that's pretty representative in most GBM tumors that you know of?
Yes. So based on what's in the literature and from what we understand, Olig-2 is in every glioma, and GBM is the most malignant of those.
Okay.
Now, the challenge is it's not in every cell in the tumor. Okay. It's in the rapidly dividing cells that are sort of at the tumor margin. So I kind of think of it like a supernova, and the tumor is kind of, going like this. And at the edge, the leading edge, are these immature cancer stem cells that are now growing and rapidly dividing. And that's kind of what's pushing the tumor out. As those cells mature, the cells in the center of the tumor kind of lose Olig-2 expression, but they're more susceptible to traditional chemotherapy. And that's the other thing, is that these Olig-2 expressing cells tend to be chemo and radiation resistant. So we've known from the beginning we're going to have to combine our drug with something else, because it's not in every cell in the tumor, but it appears to be in every GBM.
That's really fascinating. And I love the way you're describing it, because it gives me, and it'll give anybody who's watching a really good idea of how complicated and nuanced these tumors are. But it sounds like if you're able to break the barrier around the tumor itself after getting into the brain initially, that it weakens it enough that not only is it losing its food and its energy source to continue dividing and multiplying, but now other therapies can be introduced to maybe help break it apart even more so that it dies off. So it kind of paints a really good picture of how this could behave as part of a treatment plan going forward. And that's really, right, that's the goal. Companies like yours is to find something that might be able to work in conjunction with other therapies. Like we do with any other cancer, it's not a one size fits all. You can't just use one therapy and this is what's going to do it. There's so many combination therapies for breast cancer and lung cancer, and even, lymphomas have multiple therapies that are used to tackle the cancer. So why not be doing this for brain cancer?
And I think that's really where we, as a company, and really more broadly, the brain cancer community has arrived at the fact that, given the heterogeneity, right, it's a really complicated tumor. It's not a single cell that just kind of keeps growing. There's all different cell types in every tumor and between tumors. And so we know that we're going to have to do combination therapy.
Right.
And over the last 10, 15 years, our understanding of that heterogeneity has really expanded pretty significantly. So now we really know, I think, much better what are the different drugs that we need to put together. And let's not look for this one magic bullet that's going to kill every cancer cell because it just isn't going to exist. The tumor is just too complex for that to happen. But if we go at it from the beginning, knowing, hey, combination therapy is going to need to work, we're going to use targeted therapies going after these different cell types within the tumor. And we've got data to show that. I mean, our drug works alone, but it works really well with radiation. And we've got with EGFR inhibitors and CDK4/6 inhibitors and a variety of other targeted therapeutics. We're going to come up with a cocktail, just like we had, like you said, in like non-Hodgkin's lymphoma, which is a 4-drug cocktail.
Yeah.
And so that's really the goal, is to figure out that right cocktail of drugs so that we can not only delay the growth of the tumor, but actually cure these patients.
Right. So as you guys are gearing up to start a phase 1 clinical trial with CT-179, you guys are starting with recurrent GBM. Can you just explain a little bit about why you guys are going that route instead of newly diagnosed?
So yeah, ultimately the goal is to get into, to be able to deliver the drug as soon as possible in the newly diagnosed patient setting. This is a sort of first in class, first in human drug. And the goal of a phase 1 trial is safety. And so whether it's GBM or any other cancer, you start with patients who don't really have any other options, right? They've exhausted all existing therapy, and so you offer them something that's, sort of, cutting edge, but is been untested. And so we start as a monotherapy in recurrent disease, but as soon as we determine what the maximum tolerated dose is, what the safety profile looks like, better understand how the drug works, then we'll move into the newly diagnosed patient population.
Is it safe to say too that by introducing it into a recurrent GBM population, that it also allows for you guys to say, hey, this is a population that has already tried other drugs that have not worked. We can already say these drugs have not worked. So now we're introducing our trial into this, and hey, this is something that's kind of moving the needle for them, or it's something that's showing an impact in a way that they didn't see with other drugs that they've tried. And so that kind of lays the foundation to move into an expanded trial with newly diagnosed and other options.
Exactly. And that's how, we do it. And regardless of the cancer, that's basically the approach.
Okay. Yeah. Because we get that question a lot. We've had a lot of people say, well, why recurrent? Why do you start with something that's, and it's hard to explain sometimes that, you have to have something to already compare it to if somebody's already tried something else to be able to say this is effective compared to what they have done before. And we've seen those questions a lot. And I think it's because of the urgency for a lot of people that are diagnosed. You've been in the field for so long, as far as, your experience in the ER and, having been personally touched by this and the amount of clinicians that you work with. What do you see as being hopeful in the brain cancer community? What excites you about the work that's being done right now?
Great question. Things have changed a lot since we started the company back in 2013. A few things. Number one, I think when I say I mean broadly the brain cancer community, our understanding of the cancer is much better with the advent of all the different omics, right? Genomics and epigenomics and proteomics. And, we understand much better what the tumor is. Number one. So that's helpful. The models are better. The preclinical models are better. We have a lot more data of which to go after or to use. Then just the focus is on targeted therapeutics. So I think there's lots of interesting other drug targeted therapeutics that will work in combination with CT-179. I like the ultrasound technology that's opening up the blood-brain barrier for drugs that can't get into the tumor. So I think that's going to be hopeful. Our understanding of the tumor microenvironment, not just the tumor cells, but the cells around the tumor cells that are helping those cells survive and that are suppressing the immune system, right? That are tricking the immune system into not recognizing the tumor. I think there's some real hope there. There's some CAR T therapy data that looks really interesting. And then lastly, there's been some success. And I think that's really changing pharma's perspective on brain cancer and the investment community's perspective on brain cancer. Chimeric's success with OCT201 and dordaviprone, right? Um, Servier's drug for the lower grade gliomas,
Yep.
vorasidenib. Um, we can now go to investors and pharma companies and say, hey, look, at brain cancer is not just sort of this graveyard of failures, because the last drug that was approved, right, was temozolomide almost 20 years ago.
Yeah.
Right. So now in just the last 18 months, there have been 2 drugs approved. Day One Biopharmaceuticals has a drug for pediatric low-grade gliomas.
Yeah.
So I think the space has changed a lot. And so all those things combined, I'm really excited because I think, at a pivotal point now where it's all going to kind of come together, and we're going to take these targeted therapeutics, these newer technologies, the capital that's needed to make it happen. And the next 5 years, I think, are going to be pretty exciting in terms of what we're going to be able to do in terms of improved survival and quality of life for patients.
That's exciting. I think that to be able to have such a hopeful message is really important, and it's really valuable for everybody to hear how much is actually going on behind the scenes, because some of this is just stuff that they're not privy to, that they're not seeing. So to hear about stuff like this, and your trial it's not in the United States yet. It hopefully will be at some point down the road, but the trial is available, or will be available, in Australia. And we've already had a number of people that have reached out regarding questions regarding it. When is that trial actually starting?
We're on track to probably start the trial in May, June of this year.
Okay.
I think the challenge will be, because it's a phase 1 trial, it's not like, hey, we're going to go out and recruit 100 patients
Right.
and get the trial going. A phase 1 trial is you just do a couple patients at a dose,
Yep.
and then you go to the next higher dose. So if patients are interested and they're in Australia, it will, we'll be posting this on our website. It'll be on clinicaltrials.gov.
Yep.
So, I'll let you know as soon as we have a definitive start date, and
Okay.
who to contact if you're interested and live in that area.
Perfect. And, we'll post all the information as it comes up. For everybody who watches this, Curtana is a partner of ours. So anytime we are privy to any information or progress that's being made, we will always be adding it to our sites to let everybody know. I will happily walk people through clinicaltrials.gov. I know it can be very complicated and nuanced for people to read through. So, but it's exciting. It's exciting to see. This is always how a phase 1 trial works. So, for people that do watch, we don't want them to panic, because you have to start small. It's a small subset, and then you grow into the next set of the phase, a phase 1B, phase 2, a phase 2B. So there's steps that have to be taken. But as it continues to grow, it'll open up the population. And then hopefully, the goal is to start partnering with sites here in the U.S. so that the trial can be offered here in the U.S. And that's the goal. And that's the goal of every trial that we do. So I'm really excited about it. I'm excited about the work that you guys are doing. I learned something new today, because I know a lot about glioma tumors, and I had no idea that OLIG2 was an expressive gene that makes up these tumors. It was something I wasn't aware of. So, we're constantly learning more and more about how these tumors behave. And it's, as heartbreaking as it is, it's really fascinating, and it's really exciting to see some of this cutting edge technology. So thank you so much for providing us this information and for joining us today. As soon as information becomes available on when the trial is actually starting, I'll make sure to post it to everybody out there. And, we appreciate your dedication and your work in this area.
Well, great. Well, thanks, Shannon. Really enjoyed talking with you, and look forward to hopefully sharing more good news about the development of CT-179 for patients with GBL.
Yeah. Thank you so much. We appreciate having you on here. And, everybody that's watching, we will have this information in the description below the video, and we will be posting this on social media we have his Curtana Pharmaceuticals bio is on our website, so you're more than happy to go there and click on the hyperlink. It'll take you directly to their website for further info. And we also have a chatbot, an AI chatbot, that allows people on the website to search what they're looking for, and information will come up if they're searching very specific things. So, we have a lot of tools at your disposal for everybody who's watching. So thank you so much, and Happy New Year. It's a great start to the new year, so appreciate you being here.
Thanks. Good talking with you.